Hi guys. Yeah, like usual, I'm gonna just wait for 2 minutes before we start. Um, we do not have a lot of people yet. All right, I'll just start sharing the screen. Meanwhile, other people can join, I suppose. Um, that's weird. Yes. Is my screen visible in full screen mode? Would someone just confirm that?
Yes, ma'am.
Yeah, okay. Thank you. Yeah, many people have still not joined, but I'm gonna just go ahead with today's session and they can check the recording. So we are in the eighth session, so we technically have only like four more sessions after this. Let's just hope to cover as much that's there. Yeah, just going back to some of these things that we discussed earlier, we discussed about the major methods and techniques used in environmental biotechnology, and also the challenges in using these techniques for understanding microbes. And one of the things that we saw was how a lot and lot of microbes that are there in the environment cannot be cultured in the lab, which was the Great Plate anomaly. We've been seeing this in the past two classes and how this affects many of the techniques that we can generally use. And another thing is the diversity, the alpha, beta, and gamma diversity in within each of the habitats that we are looking at, how specious these microbes are and how understanding all of them and understanding how to use them can be a big challenge while trying to use them for environmental biotechnology. But techniques like sequencing, like especially the metagenomic sequencing, have really helped in solving some of these problems. So this was one of the things that I had in my previous session as well, but couldn't really go over this thing in detail. So maybe we can just do that now in today's session. How can we sequence genomic DNA? This is now, at least now, it's done through Sanger as well as Illumina. Sanger sequencing is one of the earliest technologies that was developed to sequence genomic DNA, and this can be used for sequencing shorter fragments of DNA. It can't handle large amount of samples or large amount of data like Illumina next gen sequencing can. The difference is Sanger sequencing works on sequencing these individual nucleotides. Both basically work on very similar principles, but it is on the scale at which we can do this. So DNA is denatured, and in one of the strands, a primer is used, to bind. So this primer is usually the primer. So if we are doing a PCR reaction, this primer will be the same primer that is used in the PCR reaction. So if you're giving a sample for Sanger sequencing to sequencing platforms or sequencing companies, you might have to give the same primer that you've used in the PCR reaction. So that's usually this primer, and this primer will now attach to the single stranded DNA. And ATGC, different nucleotides are provided for the synthesis. There's a DNA polymerase, there's a DNA plus primer, this strand, and there are free nucleotides and also marked nucleotides. So, marked nucleotides are nucleotides that have any radio labeling attached to each of these nucleotides. So, whenever they get attached, we get a reading, and that is what is noted down by the instrument. So it records these different signals that come from these different nucleotides. So every time one of these tags attaches, it is labeled. So this is completely like random. So different nucleotides keep getting attached, and whenever let's say there are different nucleotides that are getting attached, and when a T attaches, and if it is labeled, and that signal is being recorded by the machine. And we also collect all of these samples. So depending on the tagging, these get sorted. The tagging is recorded, and they get sorted into these different vials for A, T, G, and C, whichever the tagging is, and they're all run on the gels. So depending on how many times or how many times these A or T is present in a particular fragment or a particular sequence, that will be the weight of that particular sample. So here they are run on gel, and from this the different sequences can be read out depending on where we collected it from. So, and all of this data is put together to form these graphs, which will give us the sequence of this particular stretch. So Sanger sequencing works on this kind of base calling for each of these nucleotide types. But in a complete contrast, Illumina sequencing also adds these nucleotides one by one, but there is no labeling of nucleotides, and then gel and all of these processes are not there. The DNA is denatured, and adapters are ligated. These adapters are again provided by the sequencing companies, and they are very unique to each of the samples. These adapters also give us the ability to multiplex plex samples, especially in case of metagenomic techniques, where there are multiple species genomes and multiple species DNA present in the same mixture. We can individually ligate them with these different adapters. Therefore, we can individually separate them depending on these ligation adapters. So, and then there's, I'm not going to go deep into these methodologies. What happens, you can always check some of these details about the next generation sequencing. So there are primers embedded on this flow cell, the same primers as the ones in the adapter that we have attached. So basically this adapter sequence is what is there here, and this fragments will go get attached to these adapter sequences, and then the polymerases will start synthesizing these remaining stretches. And once the stretches are full, these sequences will bend to now attach with this other. So there's a green color and there's a red color. These are two different adapters. So the red color will now attach with the red again, and like this the multiple stretches of DNA are produced. Why is this done? So that even if there is a particular gene that is of very less amounts, especially genes that are very small in size, this will help in increasing the number, so that all of these fragments will be sequenced, and we get more confidence for a particular sequence. So all of this is defined by sequencing depth and so on. Depending on how much depth we want and how much length we want, the flow cell differs and the sequencing differs, and we can get a final data output containing gene sequences of all of this. Following this, there's also an assembly process where these gene sequences have to be assembled. But technically, we can access lots and lots of data that comes from the metagenomic sequencing. We'll also see very briefly about how the assembly works in some of the next slides. So that's how metagenomic sequencing works, but the same principle can also be applied for metatranscriptomic sequencing, where instead of genes, we'll be sequencing transcripts. So transcripts is the mRNA transcripts that we get after transcription. mRNA transcripts will give an output of what exactly was being transcribed at a given particular time by a microbial cell or so on. So we'll get a screenshot of what exactly is happening at that moment, especially here in cases like, let's say, we are testing the metagenomic profile of an infection. We'll get what are all the different viruses that are currently affecting the person, how the host is responding to it, because we'll also get the transcriptomic profile of the host transcriptome as well, whatever is being produced by the host. So we'll get an entire picture of how the virus is responding, how the host is responding, and we can look at it like a live output or a live recording of what exactly is happening because of a particular microbial infection. And this is not just for a microbial infection. This the same can be done on environmental samples to see how a particular microbe is changing the environment, or how the presence of certain factors in the environment is affecting a microbe, how the microbe is responding to a particular presence of a particular nutrient. Let's say there is a contaminated site and we want to see how the microbes in that region are responding, how they are interacting with each other. All of this can be obtained depending on the question. All of this can be obtained from the transcriptomic profile. So this is how the assembly works. So this is the same for metagenomic sequencing or meta transcriptomic sequencing. Here we have a mixed community. We have the genomic or transcriptomic information from all of them. We either extract DNA or RNA, and then we do this sequencing as described in previously, like next generation sequencing using Illumina platform and so on. And we get these smaller reads. So the bigger DNA fragments or the transcriptome fragments are cut into smaller pieces, and that is what is read by these technologies. But these smaller fragments now have to be stitched together so that we can recover the whole sequence. So that is done by something called assembly. Transcriptomic or genomic assembly can be done using a lot of bioinformatic tools, like there are packages available like Trinity, which can help you do this transcriptomic or genomic assembly. Once we assemble all of these shorter reads, we get something called as contigs. Again, contig is not a full gene sequence. It is again some of the shorter fragments stitched together. Now these contigs are assembled over one another, and we can group sequences or depending on how they align with each other as contigs. If multiple contigs align with each other, they can be grouped together as a particular sequence, or as a sequence coming from a particular organism. And we note down how much of the gene has been covered and what are all the sequence characteristics, like what nucleotide is repeated often and so on. And after this we do something called binning, which is, like I mentioned, grouping these sequences depending on how well they align with each other. And finally, after we group them, we'll be able to get these information on what microbial community is present, what sequence it is expressing, and so on. So that's very briefly and very simply put how the transcriptomic or the genomic assembly works, and how that can be used to retrieve back the information about different populations or different microbes in a mixed community. So that's mostly it. So we come to an end of our sessions on microbial techniques. Microbial analysis methods, microbial ecology methods. So if you guys had any questions up to this point, I can take those questions before we switch back into another new topic, that is bioremediation. We have spoken about bioremediation previously as well in some of the sessions, but there are some detailed slides on bioremediation and how it works and what are the challenges and so on. So before I switch to bioremediation, maybe if you guys have any questions on any of these techniques, we can quickly go over them before I switch to bioremediation. Any questions? Anyone has something to ask? I think the people who generally ask questions are not there today, I suppose. Anyway, I'm gonna just continue with the next part. My full screen still visible, right? Can someone just confirm that?
Yes, ma'am.
Yeah, okay, thank you. Yeah, so the next thing is bioremediation. So we already looked at like the definition and all, but just to briefly go over that again. Bioremediation is a managed or a spontaneous process. So why do we call it as managed or spontaneous? Because managed would mean that naturally that wouldn't have happened, but we are inducing bioremediation in a given zone. But a spontaneous process sometimes naturally happens that certain microbes are there in a site, and it clears up that site automatically. So that's how people would have actually discovered bioremediation as well. It could be a managed or a spontaneous process in which a biological catalyst, the word bio remediation itself means that the bio indicates the catalyst here is biological. It's not of organic or inorganic origin, it's not of chemical origin, it is a biological origin catalyst. Either could be a bacteria or fungi or plants. So bioremediation does not have to be just be from microbes. It could be done by plants as well. In many regions, especially the spontaneous bioremediation is carried out by plants, and it should act on environmental pollutants. These are mostly man-made or human activity derived, but it could also be environmental pollutants from natural processes like volcanic eruptions and so on, heavy metal contaminations in the soil, greenhouse gases or wastewater. These are some of the environmental pollutants that the biological catalyst acts on. To eliminate them from the site of contamination. So it does not just control or something; it completely eliminates it, and in most of the cases converts it into a non-harmful product. So that's when we call the process as bioremediation. This is what the mechanism of bioremediation is. So we analyze what are all the contaminants present and we choose microbes. So this would be a managed process, and the microbes are examined, cultured, and placed in the polluted environment. So whatever we did in the past few sessions is basically trying to analyze the contaminants and choosing the microbes, and looking at the microbes under cultured conditions and see how we can manipulate them using these different techniques that we saw so far, the sequencing techniques and classifying techniques and characterization techniques. And these microbes consume all the contaminants. So we are introducing them into the polluted environment, and we saw some of these things as well. Like we understood how the microbes consume some of these contaminants to convert them into organic nutrients, and also use them as a source for their own development as well. So we looked at how to understand these things and characterize these metabolic pathways in microbes. And these microbes then convert these nutrients into carbon dioxide and water, and eliminate these contaminants from the site of occurrence. Bioremediation, like I mentioned, need not be only from microbes alone. It could be done by plants, it could be done by fungi, it could be done by bacteria. Phytoremediation usually is done by plants, where they stabilize or reduce or restore contaminants in sediment, soil, and water wherever they are found. Bacterial remediation is done by bacteria either under in situ or ex situ conditions. Either we put them directly on the site of contamination, or we take out the contaminated portion, treat it with microbes, and release it back. Especially in case of wastewater treatment, it is ex situ, where we take the wastewater, treat it with microbes, and then release the water back into the environment. Mycoremediation is done using fungi, and it's more or less similar to bacterial remediation. It's just that instead of bacteria, we use fungi in this process. But the most important thing about either the microbe or fungi either the microbe or the fungi is that it needs to be something that can use the contaminant or the contaminated pollutants as the electron source. It should be the electron donor. It should take the contaminant and use it as an electron donor, as an electron acceptor, as well as a carbon or nitrogen source. So basically, for we have earlier also seen that for a microbial growth, we will need a carbon source and an electron source. So the microbe should be able to use the substrates in the zone of contamination itself as electron source and carbon source or nitrogen source, and under appropriate temperature and pH conditions, it should be able to convert it into less harmful products and release carbon dioxide and water. That is the main criteria of how we would choose a microbe for a particular contaminant site. So there are two things. So like I said, it could be managed or spontaneous. So in case of management as well, there are two different ways in which bioremediation can be carried out. It could be biostimulation or bioaugmentation. Biostimulation is a process where we add water and nutrients, and we add the carbon and the phosphorus or nitrogen sources, these different sources, and in the contaminants and the right, someone's yeah. So bioaugmentation, sorry, biostimulation, where we just have the contaminants and the indigenous microorganisms. So these microorganisms are already present in the site or environment where we are looking at cleaning them. So this biostimulation involves addition of water and nutrients and other sources, so that it stimulates these microorganisms that are already present in the environment to convert whatever contaminants that are there in the soil or in the wastewater or and so on. So it should be able to convert whatever is already present with the help of the nutrients that we are providing. So this is like stimulating the microbes to start acting on the contaminants that are present in a given zone. Whereas bioaugmentation is a process where we externally add microorganisms. So these are not indigenous microorganisms, they are not already present in the site of contamination. So here you can see that there's a yellow color microbe and a green color microbe. So the green color is what is already present in the given site, but this yellow color is what they are externally adding. So this is bioaugmentation. Augmentation means addition. So we add these external microorganism sources to the contaminated site, so that now the externally added microorganism can start chewing up the contaminant product. So that's the main difference between biostimulation and augmentation. But the most important thing to take care is whatever the products that are now going to be produced by these externally added microorganisms should not start becoming a problem for the indigenous microorganisms, because we know that microbes also interact with each other. We have also seen a very detailed information on how microbes interact with each other, how there could be yeah, positive interactions and negative interactions, how the products produced by one microbe can be used as a carbon source as or an energy source by another microbe. All of these factors should be taken into account, especially when bioaugmentation is conducted, because we should not end up creating byproducts that are harmful for the indigenous microbes, because these indigenous microbes are also involved in other biological processes that support the ecology. Since their niche is completely different, we should not introduce a new microbe that will now affect the entire ecosystem. So that is something that needs to be taken care of when bioaugmentation is performed. But in both of the cases, the final target is the same. We want a contaminant free environment. All of this contaminating substance should be degraded and converted into useful products that can be used by the microbes. And the best way would be to use a microbe that will convert this contaminant into something that now these microbes, as well as the indigenous microbes, can thrive on. But there are also processes where, especially in case of ex situ bioremediation or ex situ treatment processes, where the augmentation is not done in situ. So ex situ, in situ just means that in situ means in the given site or whatever is the target site, or that contamination site. Ex situ means taking a part or taking a portion of the contaminated soil or water and treating it at a different place, especially in case of wastewater treatment, like I mentioned. They add this external microbes which can treat them, but they also remove the microbial microbes and the microbial biomass from the treated output before releasing it back into the environment. So that can also be done if we are using a microbe which cannot really thrive in an environment with the indigenous microbes. So that's the two different ways bioremediation can be achieved: bioaugmentation and biostimulation. Now, so this classical example that we have been seeing from the beginning, and we have also read this in many other places as well. Pseudomonas frutida, which was engineered by Professor Anand Mogan Chakrabarti to degrade oil compounds. So there are different strains of this Pseudomonas frutida bacteria which have been used. Now, so here in this image, we can see that there are at least like 5 different strains, and a plasmid is introduced into these native organisms, and that is what now helps them. Sorry. Now helps them in degrading these different contaminants like camphor, octane, xylol. All of these are different components. They are also components of oil spills, usually naphthalene, and these can now be used for degrading these different oil products. So this engineered stain, the superbug, which is the Pseudomonas purida strain G, which actually has multiple plasmids of many of these different plasmids, and hence it is a superbug because it can degrade many of these contaminating oil components. This is also one of the strategy when choosing a microbe. We should be able to engineer it in such a way that it has a broad spectrum for the things that it can be used on. So here this same strain can be used for treating these different contaminants in the environment. So the strain is the chosen microbe is good if it has a wider spectrum for treating these different contaminants. So bioremediation, what are all the processes that are currently happening, and what are all the challenges that are mainly there in bioremediation? The most important thing is how we are going to do it. So the in situ and ex situ processes, like I mentioned, bioaugmentation, simulation, and so on, are usually done under in situ conditions. Then we should know how this will interact with other microbes and how it will affect the existing environment. So that's the most important thing that we should know. We should also know how the plants that are there in the given environment might get affected because of the microbes that we are introducing. And another major challenge is the diversity of the microbes itself. So there are so many different microbes. How are we going to classify, cultivate, and understand each of their interactions and their metabolic processes? So that's one of the problems of environmental biotechnology, and the same problem is there when we look at bioremediation as well. And Yeah, and also analyzing large volumes of data, especially genomic, proteomic, and metabolomic data is usually very large. So handling such large data and getting out information specifically for bioremediation has also been one of the challenges in this process. So yeah, so today I do not have much information. So this is pretty much it. Is what is there for this week's class on bioremediation, as well as yeah, bioremediation and the methods. So we also briefly went over how NGS itself works. So if you guys have any questions or any doubts on how the NGS works, we can probably revisit that and go over that in detail. Are there any questions, doubts, clarifications? Anyone wants to ask something? Maybe I'll just go over the NGS in a little bit more detail now that we still have time and there are no questions. Yeah, so like I was mentioning, there are these smaller DNA fragments that we get after denaturation of DNA. The genomic DNA is sonicated using ultrasonication so that it can be made into smaller fragments. Usually, the fragment length should be somewhere between 200 and 700 base pair long fragment sequences, and we attach these sequences called adapters. So this process is called as library preparation. So we are preparing a library of these smaller fragments from our whole genomic DNA. So we do a genomic DNA extraction, and then we denature them and then attach this library. So usually, even when we do this in the lab, just doing a genomic DNA extraction and mRNA extraction is sufficient, and most sequencing companies also perform this library preparation. But this can also be prepared with these kits in your own lab as well. We just have to attach these smaller adapter sequences. They are ligated using ligation principle to these DNA fragments. So they don't have to be specific like a particular primer, like how we use primers in this Sanger sequencing. These are just adapters that are just ligated to the ends of any DNA fragment whose sequence we do not know. And after that, something called cluster amplification is done. So this each of these chips are called flow cells because they have these complementary adapter complementary sequences already embedded in them. So this green portion and red portion is already embedded in this flow cell, and they'll be in very large numbers. So usually these reads, whatever we do, these reads usually are in millions. So it's like 50 million, 10 million, and so on. So these flow cells will have several of these smaller adapter sequences attached to them. So when we add this DNA fragment along with this, the adapters will bind to the complementary regions, these smaller oligonucleotide regions, and they attach, the DNA fragments attach. So once this attached gets attached to this green color part, let's say this is the forward strand, the reverse strand will now get synthesized, and once the reverse strand is completely synthesized, it will bend over and attach to the red color portion. So this end has green and this end has red. So this will now bend like this and get attaches itself to this red portion. So this process is called as bridge amplification, where the strand folds over and the free to the free nearest oligo, and the complementary DNA strand is synthesized. So from this end, the other strand will be synthesized, and when the synthesis is over, this bridge is denatured. So now they again become like this short single standard stretches. So and this process keeps happening because, like I said, there are millions of these smaller oligos attached all over the cell. So they'll keep bending over and keep attaching to the neighboring ones, and they'll form these cluster like populations. Why are we doing this? Because especially in case of transcriptomics, if we are looking at the expression profiles of certain genes, not all genes will be expressed at the same amount. Some genes will be expressed at very low amounts but would still have some reaction on the overall proteome and so on. Or it could also help us in understanding what genes are getting upregulated, what genes are getting downregulated, especially in cases where we want to understand how the microbe is interacting with the environment. Let's say we have the microbe in an uncontaminated environment, and it's a crucial microbe which is important. Let's take for example denitrifying bacteria. And these denitrifying bacteria are present along with plants. They help in plants in fixing the nitrogen. But now some contaminant in the site where the plant is found is also affecting the microbe. But to know how exactly it is affecting the microbe, we can look at the metatranscriptomic profile. We can look at the transcriptomic profile of the microbes in that contaminated site, and also that of the roots of the plant, to see if something is changing at the plant itself because of the contaminant, which is what is affecting the microbe. So we will do an mRNA extraction from these different things like the soil, to understand the microbes from the plant roots. And we will do a metatranscriptomic sequencing where we will amplify these different genes, and we'll form these kind of clusters where even the genes that are expressed in very little amounts are also visible. But these number of clusters or the number of sequences that are formed will be directly proportional to the expression. So if there are let's say 5 sequences that are there for a particular gene which is overexpressed or upregulated because of the contaminant, and there are some genes that get suppressed or lower expressed in very less amounts, or down regulated because of the contaminant, that will be maybe present as two numbers. So we will get that exact output from the sequencing as well, that we'll get 5 sequences for the upregulated gene and 2 sequences for the downregulated gene. And by counting this expression numbers, we'll be able to determine what exactly is happening because of a particular contaminant. So that's how next generation sequencing works. So yeah, once this fragments are generated, these are cleaved off, like the reverse strands are cleaved and only the forward strands are left, and it is sequenced. The forward strands, each of these is sequenced. So after each round of amplification, the lasers are passed over the flow cell to activate the fluorescent label on the nucleoside bases. So each of the nucleotide that gets attached to these sequences, they have this fluorescent labels, and when the laser passes on them, the fluorescence is activated, and we can see fluorescence for each of the nucleotide, and that is recorded as a signal by the detector. And it is recorded, and for each base a different color fluorescent labeling is used so that we get the individual record accurately. And when all of this is recorded, there is also something called paired end sequencing, where we sequence both the forward strand and the reverse strand in both the directions, so that we get information on both the forward side and the reverse side, and they are paired together to get more confidence or more information about that particular sequence, or about that particular gene. So that's yeah, in detail of how NGS sequencing works. in the same way, after we get all of these reads, so all of these reads are generated through those fluorescent signals. They are recorded, so they have something, there are something called reads. So they'll be very short fragments because we fragmented them as 200 to 700 base pair, but genes are obviously much bigger than that. So we get these smaller reads, which are then assembled together by using an assembling mechanism. So this assembling mechanism is something like, you can think of like a jigsaw puzzle, where there are like these smaller portions of the puzzle, and the whole sequence or the whole length of the gene is the full puzzle. So this assembler now brings these smaller portions together. So like how you would assemble a jigsaw puzzle, right? Like you will see that there is some edge here which matches with the edge of this piece, so let's put these two edges together, and that's how we connect this jigsaw puzzle. So that's exactly how an assembler also works. It tries putting the things together by looking at matching sequences. So let's say this is a read and this is a read, and these two regions match in this read. So it brings these two things together and put this. So now if you read the whole thing, we get a sequence stretch from this end to this end. So like that, it starts putting together smaller fragments together so that it can stitch the whole sequence. And once we have the whole sequence, we can now look at what other sequences are also similar to them, and we can start clubbing them together. We have also attached different adapters, so using adapters also we can differentiate what came from which sample or which population, and now we'll be able to bin all of them together to make them into these different. Let's say we are like collecting same color balls and putting them into a box. It is very similar to that. So all the similar sequences are collected together and put together in a box. So now we know that this sequence came from this organism number 5, and this sequence came from organism number 2, and so on. So this is like a microbial community, and once they extracted DNA, you see that some of them have a bigger size. So that would mean that probably their genomic DNA size is bigger than another one. So by putting them together and assembling all the sequences from their genome, we'll be able to map back and say which came from which of the microorganisms. So which organism is present, and if this was done for transcriptomics, we can also say which organism is happily thriving, which is overexpressing genes, which is getting down regulated, and so on and so forth. So that's pretty much what it is about um, next generation sequencing. So if you guys have any questions, anything that's not clear from any of the two things that we discussed, bioremediation as well as this. So hope it's clear what is biostimulation, what is bioaugmentation.
Ma'am, hello.
Yes, yeah, hi.
Um, Ma'am, can you please explain bioremediation in this? Because I joined late today, so
Yes, sure,
could
yeah.
not attend.
Yeah, no issues. I'll just quickly say that again. You can always go back and check the video, but I'll just summarize the important things that we saw here. So bioremediation is a process. It's a managed or a spontaneous process. Managed is when we do something to the environment, and spontaneous would mean that it is happening in an environment where it's happening directly because of the microbial interactions itself. So it's a managed or a spontaneous process in which a biological catalyst acts on environmental pollutants to eliminate the environmental pollutants from the site of contamination. And the criteria would be that the microbes itself should be able to use the carbon, nitrogen sources, electron donor and acceptor sources, all of this from the contaminant site itself, and it should convert it into products which can be which are harmless to the environment, or it can be degraded further into harmless products. So these are some of the criteria in which you would select the microbe or the biological organism. So why I'm stressing that it could be any biological organism, because in case there are also cases where it can be plants which do the bioremediation, it can be bacteria, it can be fungi. and the bioremediation can happen under in situ conditions or ex situ conditions. In situ would mean that it is happening in the site of contamination itself, whereas ex situ would mean that we are collecting the contaminated soil or water and treating it in a different region. So usually wastewater treatment is done as an ex situ process. There's also bioaugmentation and biostimulation, which is two different techniques through which bioremediation can be performed. Biostimulation is stimulating whatever is already there in the environment. If there is a contaminant and there are some indigenous microorganisms, we stimulate them by adding external nutrients or carbon sources, so that these microbes now start degrading the contaminant that is present in a given site. Whereas bioaugmentation would mean that we add microorganisms externally that are not there in the given site, that are different from the indigenous microorganisms, and now wait for these additional microorganisms to chew up the contaminants in the given site. But at the end, the result should be that the environment should be contaminant free, and whatever the products that are produced by these microbes that were initially added should not be harmful to the environment or should not be harmful to the other indigenous microorganisms present in the region. But we can also remove these microorganisms that we have added through bioaugmentation before releasing the whatever the contamination cleared product into the environment. So we also looked at how Pseudomonas fetida, which is a strain of bacteria which was engineered by introducing different plasmids. So plasmids are extracellular DNA. So this is the genomic DNA, and these circles indicate the plasmids, which are extracellular DNA. So we put our gene of interest, and by giving this plasmids to the bacteria, now the bacteria will start expressing these genes as well. So bacteria usually have plasmids to transmit information between each other. Unlike higher order organisms like plants and animals, most microbes reproduce asexually. They do not have sexual reproduction where they can exchange genetic material between each other. So they exchange genetic information in the form of extra nuclear DNA material such as plasmids. So these plasmids can be exchanged between each other through to their other openings like pili and so on. So they transmit that plasmid from one organism to another through a process called horizontal gene transfer. It can also be released to the environment, and it can be taken up by another bacteria from the soil or wherever these microorganisms are present. So these plasmids are usually engineered using genetic engineering principles. Again, when we discuss about restriction enzymes play a very important role in genetic engineering. They changed the way we look at genetic engineering once restriction enzymes were discovered. So restriction enzymes now help us in cutting these plasmids, and ligation can be used for ligating sequences of our interest. So this different plasmids carry the genetic information to degrade camphor, octane, and different oil components. So these different plasmids were brought together into this one strain called the superbug strain, which is the Pseudomonas fetida strain G, which now has these various plasmids, and this strain has the stability to express all of these plasmids together. Instead of having these 5 6 different strains to clear one site with these different contaminants, now we can use this single strain which can degrade these various contaminant substances. So Pseudomonas fetida was engineered to degrade oil components by introducing these different plasmids of interest. And we know that Pseudomonas fluorida is a classic example of bioremediation. And how are we able to do these different things? Because we have previously learned that we can understand their characterizing by characterizing their metabolic pathways, by characterizing their gene sequences. We'll be able to get an idea about what product they can degrade, what products they can take in as carbon source, and give out products that are harmless and produce carbon dioxide and water. So we also looked at some of the challenges. These challenges are basically all of the things that we learned so far. The diversity of the microorganism itself could be a challenge for bioremediation, because we know very less about a lot of microorganisms that are out there, because we have learned that many of them are not culturable, so we do not understand about a lot of them. And also, when we get this large amount of data from these sequencing techniques, assembling the information to get exactly what we want with respect to bioremediation itself could be a problem. And not doing some extra harm by introducing a microorganism is also the most important thing that we need to keep in account when we are carrying out a bioremediation process. So that's pretty much a brief summary of what we looked at for bioremediation as well.
Thank you, ma'am.
Yeah. Someone else had any other question? All right. Okay, if there are no more questions, then we can close today's session. Thank you all for joining today. We still have four more sessions. I hope you guys will continue to attend the remaining four sessions as well. And I hope all of you are preparing for the exams that will be there after the end of the course. All the best, everyone, and thanks a lot for joining.
Thank you.
Thank you. Thank you, ma'am.
Thank you.